All right. Good morning, everybody. I'm just gonna give everyone like another minute or so to make sure they're all connected and ready to go. But today I have the pleasure of introducing Sean Brockman, who is going to talk to us all about specialty soil testing and alternative amendments for soil. So Sean runs a company called The Ground Up that's based here in Colorado that covers these topics and more broadly, healthy soil solutions. So I'm very excited to have her here to speak to everyone to talk about soil health and what we can learn from her.
Thank you. Do you want me to wait a little bit longer to let people join, or do you think we should get started?
Yeah, today we also have a Spanish interpretation. So if our interpreter Valentina wants to have like a minute or two to introduce herself, and then we could get started. That'd be great.
Thank you. Hi, everyone. Good morning. My name is Valentina, and I'm here on behalf of the Community Language Cooperative, and I'm here to provide and guarantee your commitment to language justice. Language justice is a type of simultaneous interpretation that seeks to create spaces in which no one language is dominant, spaces in which everyone can participate in the language of their heart. So to achieve this objective, we will be using Zoom's interpretation function. So what you will see in a couple of minutes is that a new little button that looks like a globe will appear on the bottom bar of your screen. On that same bar where you currently have the buttons for the microphone, for the video, for the chat, etc. It'll be towards your right hand. Once you see this new little button come up, go ahead and click on it and select either English or Spanish as both the language that you will be listening into and speaking into. If you are joining the meeting on a phone or on a tablet, you will find the same options for interpretation by clicking on the button with the three little dots on the lower right-hand corner of your screen. This button reads More. Hola a todos, a todas y a todos. Buenos días. Mi nombre es Valentina y estoy aquí por parte de Community Language Cooperative y estoy aquí para ofrecerles y garantizar su compromiso con la justicia del lenguaje. La justicia del lenguaje es un tipo de interpretación simultánea que busca crear espacios en los que no domine ningún idioma, espacios en los que todos puedan participar en el idioma de su corazón. Para lograr este objetivo vamos a utilizar la función de interpretación de Zoom. Lo que verán en un par de minutitos es que aparecerá un nuevo botón que parece un globo terráqueo en la barra inferior de su pantalla, en esa misma barra donde ahorita tienen los botones para el micrófono, para el video, para el chat, etcétera. Va a estar hacia su mano derecha. Cuando vean este botoncito aparecer, hagan clic en él y seleccionen español o inglés como el idioma en el que desean escuchar y el idioma en el que desean participar. Si usted está en esta reunión por medio de una tableta o de un teléfono, encontrará las mismas opciones para la interpretación haciendo clic en el botón que tiene 3 puntitos en la esquina inferior derecha de su pantalla. Ese botoncito dice More o Más. A quick reminder for those of you that do prefer to use English during this meeting, Spanish is 20 to 25% longer than English, so please be aware of your speed. And if you have any questions or any issues whatsoever with this technology, please let the whole group know by means of the chat box so that we can work together to address them. And also, everyone should select a language regardless of whether they speak English or Spanish. Only those of you that are fully bilingual can choose to ignore this service. Un breve recordatorio es que la interpretación es simultánea, entonces cuando participe, por favor mantenga un ritmo de conversación para que yo pueda comunicar su mensaje completo en el otro idioma. Si tiene alguna pregunta o algún problema con esta tecnología, por favor comuníqueselo al grupo completo a través del chat para que lo podamos resolver en conjunto. Muchas gracias por tenerme por acá. Podemos activar los canales de interpretación ahora. Thank you so much for having me here. We can go ahead and start the interpretation channel.
Thank you. Great. And I will just ask any of the co-hosts to let me know if you do see anything in the chat and need to get my attention in case I'm not paying attention there. Well, thank you so much for having me today. And I'm really excited to be here to talk about specialty soil testing and alternative amendments. This is something that I have noticed come up in soil health conversations quite a bit. And I think that it's an important topic for us to address. I'll start by giving you a little bit of background about me and my company. So I have a company called The Ground Up. I have a background in soil microbiology and soil health. I'm also a master gardener and a certified compost operations manager. And I serve on my local conservation district board as well. So I am familiar with the soil health program and with working with producers who are using the soil health program. And so this has been something that we've been talking about a lot in using those incentive dollars to explore your soil health more broadly in a biological way. And so my company, The Ground Up, consists of arborists and microbiologists who have additional certifications in permaculture and gardening and composting. And we specialize in soil food web microscopy. We also help to create soil health plans that incorporate custom crafted compost extracts based on the biological needs of the soil. So when we look at the characteristics of soil, we have the physical, chemical, and biological characteristics. And I know everyone on this call is pretty well versed in soil and soil health, and that's why you're here. So I know I don't need to go into all the details of soil health, but I wanted to make a quick reminder to you all that the definition of soil health is the continued capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans. And it's important that we understand all of the physical, chemical, and biological characteristics of soil in order to quantify soil health. However, in our routine soil analyses, we look at the physical and chemical characteristics of soil very in depth, but we don't often get a complete assessment of the biological characteristics of soil. But the biological characteristics are very important to quantifying soil as a living ecosystem. So the services that I provide and that I am here to tell you about today are really looking more at the biological aspects of soil. So if you look at a standard pie chart of soil and a healthy soil, you should have about 1/4 air, 1/4 water, just under 50% minerals and plant parent material. And then you have that little tiny sliver of organic matter, which might be 5 to 10% if you have a healthy soil. Most typical soils in Colorado, you're going to see that more under 5%, but our goal is to get somewhere up in the 5% range or above. And it's important to have all of these ratios in a healthy soil, but that little tiny sliver of organic matter is really where all the magic happens. And organic matter is so important because it's providing all of the food, water, shelter, all of the things for the living soil, the life in the soil. And in my opinion, that little sliver of organic matter is the magic. And it's not really magic, it's science. So I know as scientists, often we like to we like to be able to explain things. And it's really just recently in the last couple decades that we've been able to have the tools to look close enough to explain the biology of what's happening in the soil. And I just want to say, Valentina, are we doing okay with like our pace? Are you able to keep up here? Okay, cool. Thank you. So, so our soil food web is a hierarchy of biology of life in the soil. And it kind of starts right in the root zone of the plant with the decomposers, the bacteria and the fungi, and the things, the smaller organisms that break down plant matter and dead organic matter. And then the second trophic level, the predators, those are the protozoa and amoeba and flagellates and things that eat the decomposers, the smaller organisms that are breaking down the plant materials and dead organic material. And then you go up into the higher trophic levels of bugs and small animals. And all of this is driven through the process of photosynthesis. So the plant is taking the energy from the sun and the water and the carbon dioxide from the air, and through the process of photosynthesis, turning that into sugars and exudates or little cakes and cookies that it feeds to the microorganisms in the soil. And then those microorganisms take that and exchange those exudates for nutrients. And that's really how all the magic happens of life on Earth. And it's so cool and so important. And when we look at soil health, I just think it's really important that we don't ignore all of that magic, all of the biology in there. And so the importance of why we want to foster the soil food web. And so I'm going to be calling this the biology in the soil food web from now on, is because we're really finding in studies that it increases yields. It can increase the nutritional value of the plants that you're growing and the nutrient uptake of the plants, the nutrients from the soil by the plants. It can protect your plants from pests and diseases and reduce your water needs, your irrigation needs. I'll show you some ways that we've found that. It reduce your fertilizer needs, suppress weeds, and protect against erosion and help even sequester carbon and remove toxins from the environment. And I'm going to go back to this picture of the magic. This little picture here is a nematode, and this picture is what started my whole career in soil biology 12 years ago. I saw this, and this is a nematode. This is actually a predatory nematode, a root-feeding nematode that is trying to break through the cell walls of the plant and suck out the insides of the plant and kill the plant. However, that little necklace that nematode is wearing is actually a fungus that has a beneficial relationship with the plant. And that fungus gets its food from the plant and in return gives the plants nutrients. So they have a symbiotic relationship, but the nematode is trying to come in and destroy that food source for that fungus. And that fungus is protecting its food from being destroyed. So it's strangling out the nematode and protecting its plant. And I just think that that is such a great image to show exactly why the life in the soil is so important, and how if you have a healthy soil food web, it can help to do your pest control, to protect your plant, to help your plants be more, their immune systems boosted and be more vigorous and viable to grow healthy and have greater yields and need less inputs. So how do we foster the soil food web? Well, we do it at the ground up with compost extracts and compost teas. And you've probably heard a lot about these. There's big buzzwords about compost tea. It's kind of a hot thing right now. Lots of alternative amendments are coming on the market. And so I'm just here to tell you a little bit more about compost extracts and compost teas, and kind of make sure that we all are on the same page when we say these terms. So compost tea is not the leachate that comes out of your worm bin. It's not the stuff, the liquid that's seeping out of your compost pile. That's leachate. That's not compost tea. Actively aerated compost tea is a process that extracts the biology and the organisms from a healthy compost to make a liquid solution that can be then applied to plants and help to inoculate the soil with biology. This picture here is a pretty cool picture. This is from a plant trial, a turf trial that was done at Rodale Institute in 2012. And you can see the root growth and the soil, the aggregates that have formed around those roots that's just 1 year of doing compost tea on the handful, which would be your lookers left, compared to conventional fertilizer on the right. So it's pretty incredible to see when you foster biology in the soil, it really helps to form those healthy soil aggregates and those root systems. Here's how I make compost tea, and there's a few different ways to do it. And so I'll show you some different tools that can be used, but just to give you an overview of how it's done is you have this extraction chamber, and it's essentially a large tea bag in a big vat of water. And so you have the water and you have the extraction chamber that has the dome, and the dome has a big tube in it that's going to be blowing air out into the tea bag. The tea bag is filled with the compost, And the air agitates the compost particles and releases the microorganisms from the compost particles so that they can swim out into the water. And that's how you get the concentrated liquid extract from the biology that came off of the compost. The air is really important in there. And in this system, I also have another, you can see this big pipe. I don't know if I can show you this big pipe kind of coming down here behind the tea bag. That's also air that is circulating air into the water. And this actively aerated state is very important to help maintain healthy microorganisms and not breed any anaerobic species. We want to make sure that we have a healthy product that's safe to apply to the environment and safe for human health. So we're not breeding any anaerobic bacteria and or microorganisms. And so we make sure that we have an aerobic environment in that. Here is some equipment. So you can just see this GOT brewer is the brewer that I use, but there's also cone brewers that you can find. Those are really nice, easy to clean. When you're looking for a brewer, you really want it to be very easy to clean. And cleaning is a very important step when making compost tea. You don't want any biofilms to be accumulating and creating anaerobic pockets that could then release those anaerobic organisms into your liquids. There's also a big, you can see the inside of this other brewer here to see how easy it is. The stainless steel is very easy to clean with high pressure water and cleaning all of your equipment. And then I'm also showing you some application equipment here. There's different ways to apply the equipment. And we have a big truck with a tank on it and a pump and a hose. We also have a side-by-side that has a smaller tank on the back with a boom sprayer. Works really well on a lot of the hay fields and the agricultural spaces that we work with. I've seen it on golf carts, which are really easy. More people like that if they can get an electric golf cart, a little more environmentally friendly option. And you can also do as small as a backpack sprayer as well if you're just doing smaller spaces or hard to get to spaces. Also, drones are coming on the market, and lots of people are doing the drone thing with the compost extract as well. The drones can't hold as much, so I'm still working out the cost benefit analysis of using drones for this. Here's just to kind of show you ways to apply compost extract is you can do root injections and inject it directly into where the root zone of, we're using that for trees a lot. And we've been doing some orchard trees, which is really effective in getting it where you want it to be. Soil drenches, foliar applications, you can put it right into your irrigation system and your boom sprayers. And a couple considerations on application would be that you really need to pay attention to the filter size. If you have a filter, removing the filter altogether would be best. And if you can't do that, making sure it's not like a metal filter. Those filters can kind of shred the little microorganisms. So you don't want to ruin all that you've worked so hard to make once you're putting it out there. Also, the pump type, a diaphragm pump is going to be the best to make sure that we're not shredding up those little organisms. And the nozzle size, we're looking at 400 microns or larger for the size of any little spaces to get those through. So we just really want to make sure we're protecting the integrity of the product during the application as well. So every step of the way, and making sure we are cleaning everything really thoroughly to make sure that we don't have any residues or biofilms accumulating. Also, I don't use the same equipment that I would use for my chemical application. So it should be totally separate equipment. If you can't get separate equipment, you would really need to clean it very, very, very thoroughly and make sure there is no chemical residue in there. So kind of going back and forth between a chemical and a compost tea and the same equipment would be really challenging and a lot of labor to clean in between each time. It might be worth it to just get new equipment for that. So I will give you a few examples of how we've seen success with compost teas. And so, sorry, the difference between a tea and an extract is just agitating those particles and releasing them from the compost into a liquid. And then a tea is when you feed that liquid to then multiply those organisms. So you might feed it with things like kelp, molasses, fish meal, things that are going to feed and multiply those organisms. You would be mindful, though, that by feeding, you are selecting for the organisms that like that food. So if you want a true extract of the organisms that are in the compost you're using, then you would just do an extract. But if you're trying to select for and feed for a certain type of organism, then you would select a food that's going to breed that type of organism. So here's some examples. This is a field out in Gypsum, where I live. I'm in the middle of Colorado in Eagle County. And we started this 4 or 5 years ago. This was just after one season. You can see on this alfalfa field, we did a compost tea extract. They graze this in the winter. They allow their cows out there for winter grazing. In the summer, their cows are out in the like Forest Service land, so like rangeland. But when they bring them home, they graze here, and then in the summer they're growing alfalfa. And now they're part of the soil health program and doing some cover crops and diversifying and no-till, and so trying other things as well. But before they got into the soil health program, we were just experimenting with compost tea, and you can see the pictures of the alfalfa. You can just see without even looking at the soil test. And this is what I'm hearing so much from the producers, the people out there in the field, just walking through like, wow, I'm looking and you can see the chlorosis on the alfalfa on the left, and on the right, it is green and lush, and they are clearly more healthy plant there. And so if you can just see that, imagine how much more healthy it is for the nutrients going into their animals as well, who are grazing on that, or they're cutting it for haying. I also have a little picture of the mushrooms that you can see because there was some cow patties in there. And so inoculating, we were seeing a lot of fungal growth on some of the cow patties under the alfalfa, which is what's really cool. Here's another pretty cool example. This is not a study I did, but a study that I researched and found. And when I was first starting this business was they used this machine, this GOT machine, the same machine that I use, and they tried to do a maintenance program at Harvard University. So the groundskeeper at Harvard wanted to check out this more organic option. And so he transitioned and did 8 months of compost tea applications. I believe it was one application a month. And in just one season, you can see the difference in the root growth and the soil, how the soil was more compact on the left and a little better structure on the right. But the really big takeaway here was 30% reduction in their irrigation. And when we are looking at water here in the West, this can be such an incredible tool, just partnering with the soil food web to get healthier soil and reduce our water, and really reduce our water inputs, retain our water in the soil. Here's some more research on soil food web being effective in reducing erosion. So we're looking at the soil food web really for response when there's fires or restoration projects as well. And you can see that the soil loss was so there was so much less soil loss when they were able to inoculate with biology in the soil in these erosion trials. So that gets into aggregate stability, which we talk about a lot in soil health. And aggregates, as most of you know, are the particles that are held together by clay, organic matter, roots, bacteria, fungi, exudates, the glues that the bacteria and fungi release to have that relationship in the soil with the plant roots. And it's really the ability for the soil to regulate the movement and storage of air and water. So aggregate stability is really important for soil health. It's our goal of what we're looking for. And these pictures show you under a microscope what it would look like, what a soil aggregate looks like. You can see the fungus and the root hairs coming out of the one on the left, and on the right, it's kind of a healthy soil. You got a healthy beneficial nematode, lots of biology in there, and some soil aggregates around. So that's what we're looking for when we're looking under the microscope. So something else that we do at the ground up, and you're seeing a lot more of this around, is looking at soil through a microscope. And so the microscopy is a great way, a great add-on to your soil health test. So in addition to all the testing that you're doing, when I mentioned that our routine tests kind of only scratch the surface of the soil biology, this is one way to look closer at what is in your soil. And so we really look at quality when we're making a compost extract, when we're looking at the compost we're using to make our extracts, we're looking at it under the microscope. We're sending it into the lab. We're doing all of the testing, and you're going to get some resources as well today about some other there was supposed to be another presenter today who couldn't make it, but we'll make sure to get that resources out to everybody. I'm not the only person doing this. There's a lot more resources. I'm just here to tell you about the tools that are available to you. So there's lots of additional testing and resources that you can do. And microscopy is one of them. So we're really looking at the quality of what we're putting in. So not all compost is created equal. And I think that's important to note. I've done a past presentation about composting and compost application, so you can look back in your webinars and find that one as well and learn more about compost. But I just think it's important to note that there's many different ways to compost. And as long as that compost is reaching processes to further reduce pathogens and protect human health and the environment, then we are making products that are for every intended use. But note that not all compost is to be used for all. It's not meant for every single use. There's different types of compost, and there's different uses for compost. So know what is your use for compost? What are you trying to accomplish? What are you trying to achieve? And find the compost that meets that use and that meets that benefit. So in particular, we make some specialty composts, and you've probably heard the big rage around fungally dominated compost. So we've been doing some experiments with this, and I've run large scale compost facilities. I teach composting all over the U.S. I'm going to say that composting is an incredible tool that we have at our disposal, and it's really fun to experiment with different types of compost and then look at it under the microscope and see what are we getting out of this compost? What are we trying to select for in making these composts? So we've made this fungally dominated compost. It's the Johnson Sioux pile. This is kind of what it looks like. You have air pathways. This one isn't reaching temperature, so it's really important that we're looking at it under a microscope and sending it into the lab to get tested to make sure that it doesn't have any pathogens. But we are seeing incredible fungal activity. And so I show you here some examples what it looks like under the microscope to see this kind of fungus. You can see the close-up view of the fungus under the microscope. And then below that is a little bit, that's the 10x. So a little bit further away of these long hyphae fungal strands that are holding together. It's holding together the aggregates and the soil particles. And in addition to that, fungus is mining more nutrients. It extends the plant root and is able to do so much more for that plant by having relationships with fungus in the soil. So really cool stuff. So when we make a compost like this, we can make an extract that is a fungally dominated extract because we now have the compost to be able to pull that out. We can also make extracts then to inoculate maybe like a commercial compost or a bigger compost that hasn't gone through the life phases long enough to have the population of these organisms repopulate. And so we can kind of speed that process up by inoculating with that kind of compost. And we also have worms. So we've got worms where at the ground up, and we do vermicast. This is a flow through vermicomposting system. So you feed the top, You can see on the left what our flow through system looks like. You feed the top and you harvest from the bottom, and the worms kind of all crawl up, and then their castings go down. We do pre-compost material before we put it into the worms. Not everybody does that, but the worms really have made some incredible compost. And in looking under the microscope at the worm castings, we are seeing the most diversity of life that we have seen from anything else we put under the microscope. Different types of compost and soil. This is the magic. So worms are awesome and really cool. So if you can get your hands on some worm compost, definitely get it tested. Make sure that we are breeding something that's healthy, and that, especially if we're going to make a tea out of it and breed those organisms, we want to make sure that what we're breeding is healthy biology. So going back to the soil food web, which we always go back to the soil food web. It is very, very central to everything we do in soil health. I was just going to take a little time to kind of go through the soil food web, but I think I'm going to take a pause and let's see what kind of questions before I kind of dive deeper into the soil food web. Maybe we just want to, oh, you know what? Hang on. Let me just say one more thing about microscopy before we go into the soil food web, is that as we, let's stop and we'll take some questions now. Let's see what kind of questions we have.
Regan had a question earlier on your slide 6, the first image of your soil food web, and then you had an image of the nematode being strangled by the fungus. Regan was asking, like, what is the string between the two bacteria? I'm assuming that's the fungal strand, or is that?
Yep. With the nematode, let me look at the chat and see if I can. Okay. So that was during slide 6. So I'm thinking that's maybe this string here. Oh, you can't see my cursor, can you? No. The string around the nematode, that is the fungal hyphae. Yep. And so that is that's a fungus that is strangling the nematode. And so that's fungal hyphae, and it is, when you looked at my closer pictures, let's see, fungally dominate. So when you look at the fungus under the microscope, you can kind of see that it is building, and it kind of has segments in it. So it's building. That's why also fungus is so great at holding on to carbon, because it is building carbon in its hyphae. And so it's like building and holding and storing carbon. And so every time we till and disturb the soil, we are breaking up those fungal strands as well. So releasing that carbon. So if we really want to hold carbon in the soil, helping to maintain the integrity of that fungus and allowing it to continue to build is really important too. I hope that kind of helps answer that.
Awesome. Thanks. Yeah, I think this is a good time to call in, take people's questions, since we've got some announcements and then we've got about 20 minutes left. Does anybody have any questions for Sean? Jen, you raised your hand. Do you have a question?
Yeah, I do. Thank you for this presentation. You had mentioned about say, like when you're brewing a compost tea, certain things that you feed it might select for certain organisms. Can you share a little bit more about that and how to go about comparing, say, maybe where you're at based on like a soil sample analysis with where you want to go? Like, how do you find that information to then guide your next step forward in your compost or compost tea or whatever selection process?
Yeah. We look at soil analysis and specifically soil food web analysis that's telling us what microorganisms are in the soil, so we can we do that by kind of counting and seeing what's present. And I'll actually just kind of go to my next slide. So when we're looking at the soil and under the microscope, we're identifying the microbes by morphology, which is how they look, and mobility, how they move. And so when we are doing that, we can kind of identify what's in there. Is it beneficial? Is it not beneficial? So an example would be that on one of our clients' fields, they were having these brown spots in their fields. And we went in and did a soil analysis and we found root feeding nematodes in there. And we were like, well, here's the problem. So we had to kind of prescribe a plan to eliminate the root feeding nematodes. And then come back in. In this particular instance, we did like a nematicide. So we did bring in a company that could do a nematicide on those spots because it was really bad. And then we are coming back in this spring with beneficial nematode tea. So when you asked how do we feed to select for the different microorganisms? So bacteria really like sugar. So you might have heard about molasses in compost teas. That's going to breed your bacteria. Most of our soil already has a lot of bacteria, so we don't put a lot of molasses in our teas because we don't need more bacteria. Some soils are completely, like, not completely dead. I mean, there's a little bit of bacteria, but when you look at the biomass of the bacteria in some soils, there's like not very much. So sometimes you do need bacteria because you need the start of the soil food web. You need to kick it off and get something for those higher level predators to eat. So that would be an instance where you would add molasses. Kelp meal is going to feed some of those higher predatory level species. You also are selecting for compost that have the higher predators in them already. If you don't have a compost that has the species in it that you are trying to get, they're not going to appear out of thin air. I mean, they do in your compost. If you build it, they will come. But in your compost tea, you're really extracting what's in that compost already. So you want to make sure you select a compost that has what you need. So in an area where I have lots of trees and shrubs, there we want a higher fungal to bacterial ratio. So in our soil food web, we give you a FB ratio, and that's how much fungus do you have to the bacteria. And so we really would love to get like a 1-to-1 on your like early successional things. If you're looking more like a forest, if we can get even like a 10-to-1, that would be incredible for like a forest, 10-to-1, 20-to-1. But that's a lot of fungus. So you're going to use like a super high fungus. So for our trees and shrubs and perennial woody species, we're going to use more of a fungally dominated compost to extract and do a compost tea. And we're not going to feed it molasses. We probably won't feed it. We'll do an extract because we want to just extract the fungus that's there. If we're doing protozoa, we do a longer brew because those protozoa are there to eat the bacteria. So if you are breeding the bacteria and you have some protozoa in there, then the longer you wait, you let it brew, the protozoa are going to continue to consume the bacteria and multiply. You will get to a point where the protozoa are maxed out and they run out of air or food and start dying. So it's going to be a little bit longer, like a 48-hour brew for a protozoa. So we would do that longer if we're looking for a protozoa. And protozoa would be like flower gardens and turfs and annual species and perennial too. But looking if there's areas that need help, and they already have a lot of bacteria, they could be overloaded in bacteria, and we need to get the protozoa in there to balance out that bacteria mass. Does that answer?
Yeah, that's a great start. Thank you so much for that.
Yeah. And my next few slides do go into the biology and more in depth, but I wanted to make sure we took some time to answer questions and stuff before we go in.
Thanks, Sean. Yeah, Steve had a question in the chat on how do you determine application rates and what is the best time of year to apply?
Yeah, thank you. That's a great question. So application rates depends on the quality of your tea. So there's a lot of studies Elaine Ingham does the Soil Food Web School, which a lot of people have probably heard about. And she claims that you only need 1 gallon per acre, which is really hard to calibrate. So you would then dilute that 1 gallon, but that's 1 gallon knowing that you are doing these microscope tests, and we feel more confident about like 10 gallons per acre, 5 to 10 gallons. And we can still dilute it that way, but we're at the 5 to 10 gallons per acre on agriculture. And we're looking at every single batch under the microscope. And we know that what we're putting out there is full of what we want it to be. So whether that's a protozoa brew, a nematode, we can actually do a nematode extract that's a little bit more in depth. So I won't be able to go through all of that today, but we're trained through we have 2 people on our staff trained through the Soil Food Web. So we're really making sure the quality of what we put out there. And I think that's just the most important thing when you're looking at this stuff is make sure that whoever you are getting your compost tea from, or if you're brewing it yourself, send it in and make sure that you're getting what you're anticipating out of it. So that application rate really depends on how concentrated your brew is. Some people are doing 20 gallons an acre. I used to do 20 gallons an acre, and my fees were very low because I didn't know as much. I've been doing this for 12 years now. When I first started, I was giving away all my product because I didn't really know what I was doing, and I just wanted people to try it, and it was just really cool side project for me. And I wasn't running it like a business. I was running it like a experiment. But now I've been doing this so long, and we are really dialing in what is in there. And so now I do charge a lot more because I don't have to put as much on. So because I'm putting a lot more into every batch, all the labor and getting it under the microscope, and having a microscopist on staff and making sure that we are we're putting out what we say we're putting out, and making these specialty composts to put in. So you could also make your own compost tea, but just maybe buy the inputs from somebody who's guaranteeing that what's in there and getting that under the microscope, or DNA testing. I haven't talked about DNA testing at all, but that is definitely up and coming, and still a little bit out of the price range of your average producers, probably, but it's getting there. It's getting a lot more accessible, and we're able to have some really cool tools that are coming out, like handheld tools to be able to do DNA testing. So I hope that answered. Oh, time of year. So we really like to get a good push in the spring, especially right as the plants are starting to, like, the bud break time and the time when all the plants are starting to come out, because that's when they're really putting out exudates as well. That's when they're starting to photosynthesize, and we want to get it at that time when that plant is doing photosynthesis, so that because it's attracting the microorganisms to its root zone and through its exudates, and having those exchanges and those relationships with the organisms. So then it's fueling the soil food web to kind of continue what we're starting with the inoculation. So spring, I do heavy. I'll do at least one application. Sometimes I'll do 2 applications, 2 or 3 in the spring. You can find there's some of the studies that are being done are seeing great stuff just by 3 spring applications and no more. We usually do 3 applications a year. I'll do a couple in the spring, and then 1 or 2 in the fall, depending on kind of what my goals are for the producers, or how the starting point of their soils. Fall's a great time as well, because you're really getting the microorganisms in there right before everything kind of goes into hibernation mode for the winter. So you're getting especially if they're doing like a no-till and like leaving the plant residue on the ground for those organisms to have food all winter, that's really huge too. So depending on their production, the soil health practices that they're putting into place kind of determines what time of year that I'm doing applications, so that I can maximize those practices and what food is going to be available for the microbes in the soil.
Awesome. Thanks so much, Sean. Does anybody else have any questions? So yeah,
I've got one.
Oh yes, go ahead.
Would you apply before you plant or after you plant?
Yeah, I like to apply right after. I think it's nice to have the seed in the seed to soil contact and then inoculate with that microorganism. It wouldn't be harmful to do it before and to establish, especially if you have any kind of plant residue on the ground, that like if you're doing a no-till drill into existing vegetation, then I wouldn't say you have to wait until after the seed is planted, but if you don't really have much on the ground, it would be good to have the seed planted and do your application right after seed planting. And if you're putting it right in, I have people putting it right into their irrigation systems too. So just putting it right in with the irrigation after planting. Thank you.
Great. Thank you, Sean. This has been incredibly informative, really interesting. I do want to remind everyone, this is, if you choose to do one of these specialty type soil tests, you can claim that as part of your reimbursable expense for your participation in the soil health program. So if one of your producers wants to send their soil in or their compost in and have the ground up, or our other group that was going to be here today, which is Pure Soil. And there's
other people in the
groups,
state. Yeah.
So feel, if there's other folks doing this type of thing and you want to, or you want to send it to Sean or these other people, go ahead. And that can go towards people's reimbursable expense for their participation in the program. So that is worth noting. And yep, go ahead, Lisa.
I did also want to touch, because Sean mentioned DNA testing, that for producers in our program, the soil tests that they're getting is going to have some taxonomy results back to them. So you will get some DNA sequencing and you will get a snapshot of the soil food web that's in your soil currently when we sample it. So that can be something you look forward to as well. But I think we will send out Sean's information after the seminar, and we're going to transition over in the last 10 minutes or so to our announcements, unless there was any last final things you wanted to say, Sean.
Yeah, I just wanted to say I do have 12 more slides that we didn't get to, which is okay. I didn't plan on getting to them. They were kind of like in there as a bonus. And I think you were going to send this presentation out, right?
Yeah, we can certainly send it out.
If you're welcome to. So there's a lot more about the details of the different soil food web players, and it's kind of lined up like how I would present it. So it might be confusing. So if you have any questions on any of that stuff, feel free to reach out. I put my contact information here. And if you're looking for, somebody in your region to do this microscopy, it's really important to get it done within like 24 hours of taking the sample, and taking the sample actually is kind of a unique thing too. So feel free to reach out to us. And if we're not in your region, we'll try to help connect you to somebody who is. We just wanted to make sure that you have the information and grow the awareness and the knowledge of all of these tools for you.
Awesome. Thanks so much, Sean. Yeah, John, do you want to transition?
Hang on. One more thing.
Sorry.
I also want to say that. Sorry. I just want to also say that there's a lot of research going on too. So there's some really cool things going on with grants, and the U.S. Compost Council has gone for a big grant to try to get some more research around compost application rates in particular. And then Enriches is going for some cool grant money to explore more of this extract and compost tea throughout Colorado region. And we're just looking at other grant opportunities as well. So if this stuff really excites you, or you have producers that you're working with who are really excited about it, I would love for them to reach out to me, because I'm trying to get a list of who might be interested in doing some research projects to explore this further and get some more data. The science is really important for the growth of this industry, and we would love to see who's interested in exploring this more. We don't have any money yet, but we're going for it. So if you want to be a part of that process, definitely reach out and I'll keep in touch with you. Thank you.
Awesome. Thank you, Sean. All right, I'm going to do a few quick announcements here. First and foremost, we have made 3 hires for our soil health field specialists. I think only one of them is on this call. And that would be Vanessa McCracken, who we hired for our Northeast field specialist. Vanessa, do you want to say hello?
Sure, I can say hello. I can only see Sean's screen, so I'm sure you're all out there. Happy to be here at CDA. Really excited to work with conservation districts and other entities in Northeast Colorado and wherever else I'm needed. Just looking forward to meeting you all and spending some more time getting to know you and your producers.
Thank you, Vanessa. We also hired Reese Melton for the Northwest, and I don't see him on the call. He might be out doing something else. And then we hired Toby McPartland for the Southwest, and Toby and John Rizza, our Western coordinator, are out in the San Luis Valley today. So they're already out there getting boots on the ground. So you're going to be hearing more from those folks and seeing them out in the field, and they'll be contacting you and talking to you, and we'll be getting you some technical assistance for your producers that need it. We also are very close to hiring a person for the southeast corner of the state. So I think we're going to have all 4 of our soil health specialist positions filled with some very qualified, technically apt people that'll be helping you out as we go forward. So that was the number one thing I wanted to say. Secondly, the first round soil sample packages have left our building and went out with some of our field specialists. And we'll be getting those around to you. So you're going to start seeing those show up. So if you had producers in the first round, you'll be receiving a stack of envelopes with the sampling bag in there. Some of them go to Ward Laboratories. Some of them go to CSU Spur Campus. So you might see two different looking sets. Well, depending on where you're at, you'll see one or the other. So those will be coming to you. We've provided you with some instructions on how to get those samples taken and who to send them to. And there's return labels on the packages. So that should be coming your way. For second round soil samples, so people that we're just enrolling right now, we're putting those packages together as well. Those ones will be return postage paid. So we're going to pay for the postage for you to get those samples to the laboratory. All of those samples will go to CSU Spur Campus to be tested there. So you'll be getting more packages coming up. If you have people in the first round and second, you're going to get two separate packages. If you only have people in the second round, they'll be a little bit further behind, but we're working on those. They will also have, as well as the sampling bag and some info that we need you to send, or to some info on taking the samples. There will also be a small test tube in there with some fluid in it, and that fluid is the DNA stabilizing fluid. As Lisa said, we are going to be doing DNA testing on all of the samples this second round. So we will provide you with some guidance. We're going to have a upcoming webinar here with the folks who are in charge of that DNA sampling, and they'll provide you with guidance on how to be as clean as possible while you're taking a soil sample and get that soil into that test tube, and how much we need in there and things like that. And that DNA stabilizing fluid will kind of freeze a snapshot there, and then we can get that sent off. When you get that sent off, it'll get to the right place, and they'll do the analysis of the DNA there, as well as your regular soil analysis. For those second round folks, and maybe first round, if you have people that are waiting on us for like NPK tests, this is going to take some time. So you're better off to have those people go ahead and do regular NPK tests on their own. That is a reimbursable expense if they've gotten their enrolled practices emailed out to them. So feel free to take soil samples on your own instead of waiting for us. When you do get these ones, the ones we're doing, they're very in depth, and it's going to take some time before you get them back. Our regular soil analysis takes about 21 days from when the lab gets it to when it's completed. So you're probably not going to see the results for close to a month. The DNA, obviously, it takes a little bit. It's even going to take longer than that, but that'll be in a separate report. And yeah, so if you're waiting for us for soil sampling, or if you folks want to apply nutrients related to their soil health practice, or want to apply compost, and we're saying that they have to have a soil sample to do that, and they're just kind of on hold waiting for ours to occur, we are going to be a while. It's just going to take some time. So what they should do is move forward, get an MPK test on their own, and submit that. They can submit that with reimbursable costs. The other thing I would like to mention is our next meeting next week. We will have the CSU sociology team that's working on our sociology study here for the soil health program. They're going to be here. They're going to kind of give a brief overview of what their sociology study is all about, why they're doing that study, and what they're hoping to find out through that study. So they'll be here next week, and we'd really like as much participation as we can get with those folks. So this is really awesome to see so many people here today. Hopefully we get a good turnout next week too, to kind of so you can get an overview of what those guys are up to, and you're going to be running into them, kind of coming across some of their surveys and stuff that they're doing that they want to get out to producers. So I think it'd be helpful if everyone knew what their intentions were and what they were up to. So please attend that next week, and we'll maybe have some other announcements then as well. So anyway, thank you so much, Sean, for that awesome information, and thank you everybody for attending, and we'll see you all in a week.